And now we're going to look at the cytoplasm of the cell. This is cellular material outside of the nucleus but inside the plasma membrane. And remember I said it's kind of like jello in a bowl, the bowl being like the cell membrane. The cytoplasm is the substance basically that holds on to and supports like the organelles and the cytoskeleton and all of that stuff. And again, what is jello made up mostly of? Well, water. So cytosol is the fluid portion. And then the cytoskeleton, like I said, it supports the cell. It gives the cell shape because its structure, it's made up of microtubules, microfilaments, and intermediate filaments. And again, these filaments can do a lot of different things besides giving it structure. As you see here, it provides kind of a support for the different organelles. Organelles can move along. We have actin filaments. These are made of microfilaments, and they're composed of actin subunits. They're about 8 nanometers in diameter. And this is what can be used, as I said, for moving things in the cell. Actin and myosin are found within muscle cells, and that's what causes muscle cells to contract. And when they relax, they elongate again. But the actin and myosin together is what causes contraction. The actin and basically, other cells allows for movement of these organelles. Intermediate filaments are about 10 nanometers. They're protein fibers. What else? Microtubules are composed of tubulin protein subunits. Microtubules are 25 nanometers diameter tubes with 5 nanometer thick walls. And again, these things can be used to make cilia or flagella and so on. So just like our skeleton gives us structure and some function, cytoskeleton gives it structure and function. And then cytoplasmic inclusions, like I said, these are non-living materials that are found within the cell, like starch granules or glycogen granules or sometimes crystals, some waste products that might build up within a cell. And then organelles. Again, organelles are tiny little organs, like we have specific organs in our body. These are tiny organs in our cells, call it organelles. So they're small specialized structures for particular functions. Most have membranes to separate the interior of the organelle from the cytoplasm. And they're related to specific structure and function of the cell. Now these are the centrioles. They're found in specialized zone near the nucleus called the centrosome. And each unit consists of microtubules. And before the cells divide, these centrioles divide, they move to the opposite ends of the cell and they create these things called spindle fibers. I don't know if you've ever done a zip line before or seen a zip line. This is kind of what they create. They create these zip lines going from one end of the cell to the other end of the cell. And this is what chromosomes are going to attach to. And then when the time comes, the chromosomes split in half. Now, mind you, they've already replicated themselves. So when they split in half, you're still getting a full amount of chromosomes going to one side of the cell, a full amount of chromosomes going to the other side of the cell. But how they get to the other sides of the cells is across these zip lines, these spindle fibers. And again, those are created by the centrioles. And then cilia. We have appendages that project from the cell surface. They're capable of movement, and they move materials over the surface of the cell. And it kind of is hair-like looking under the microscope. When we get to histology, I'll show you the difference between like cilia and microvilli when you're looking through the microscope. even though for this class we won't actually be looking through a microscope, but we will be looking at slides. And the way these things move, you've got what's called a power stroke and a recovery stroke. Think about a swimmer. Swimmer stretches out their arm, that's the power stroke, and then when they pull their arm back, that's the recovery stroke. And then when they put their arm out, the power stroke, pull their arm back, it's the recovery stroke. Same thing with like a volleyball. Toss the volleyball up in the air, your arm comes back like the recovery stroke, then it stretches out into the power stroke to hit the ball. And then flagella, it's similar to cilia but longer. There's typically one per cell, and the only cell in the human body that has a flagella is going to be the sperm cell. Okay, and like I said, it usually has one per cell. Sometimes you'll get a couple per cell, but that particular sperm cell is not very functional with the two tails. So it's usually just the one tail, the one flagella. And it's going to move the cell in kind of a wave-like fashion. A little different than cilia that has that power stroke and recovery stroke. This is going to be more of a wave-like action. And if you take microbiology, you'll learn about bacteria having flagella. And it's set up a little bit different than like what our flagella is set up, or eukaryotic cells flagella. It's just a different mechanism of how it actually works. In the bacteria, it spins kind of like a propeller. In the eukaryotic cell here, it's more of a wave-like action. And then microvilli, this is extensions of the plasma membrane. So people get this confused with cilia, but they're a lot smaller than cilia. They're 1/10 to 1/20 the size of cilia. And there's going to be a lot of them. And what they do is they increase the cell surface. And you know what it kind of reminds me of? Kind of reminds me of this guy. Okay, so we've got Bart Simpson. And see how Bart Simpson, this is not hair on Bart Simpson. This is more like extensions of his skull. As a matter of fact, his head kind of looks like a columnar epithelial cell. And it's the columnar epithelial cells where you're going to find this microvilli. And again, the function of the microvilli is to increase surface area. And so what's a place where we would need increased surface area? Well, in the intestines, you're going to find a lot of these cells with the microvilli in your small intestine. And that is, again, to increase surface area so that you're going to absorb the maximum amount of nutrients. Matter of fact, in the intestines, which you'll talk about next semester, you've got surface area upon surface area upon surface area. So you have a bunch of these cells with the microvilli, their surface area. Then you have this long extension called a villus. And that is like a finger-like projection that has all of these columnar cells attached to it. Okay, and so you have millions of those, and so that increases surface area. And then there's folds within the small intestine called the plica circularis. And don't worry about these terms now. That's stuff you'll get next semester. But and that increases surface area. So you have surface area upon surface area upon surface area, again, to increase the amount of foodstuffs that you're trying to absorb, the nutrients you're trying to absorb. And next we look at ribosomes. Ribosomes are the site of protein synthesis. In other words, they make proteins. And it's composed of a large and a small subunit. And well, you know what that kind of reminds me of? A large and a small subunit? It's going to remind me of a stapler. See how the stapler has a large subunit and then a small subunit. And what does a stapler do? Well, it attaches things. Like if I stapled a sheet of paper together at the top, and then maybe went to the bottom of one of the sheets, and then the top of another sheet of paper and stapled that together, I can make a whole long chain of paper, right? Well, that's exactly what ribosomes do, is they take amino acids and they staple them together. Okay, they don't staple them together, but they attach them with peptide bonds. Okay, so they kind of act like a stapler. And they will make this long polypeptide chain. And then when you take that polypeptide chain and you fold it precisely, that's a protein. Okay. And there are a couple of different types of ribosomes. There's free ribosomes, and that means they're not attached to anything. They're kind of floating around in the cytoplasm. Or they can be attached to endoplasmic reticulum. And when we attach them to endoplasmic reticulum, it's going to give it a certain texture. It's going to make it look like sandpaper. And what's the texture of sandpaper? It's rough. And so this is where we're going to have our rough endoplasmic reticulum. And so, again, the rough endoplasmic reticulum has attached ribosomes, and proteins are produced and modified here. So that's the function of rough endoplasmic reticulum, to make proteins. How do you know that? Should have known that even before I came to this. I should have asked you what the function of rough endoplasmic reticulum was. Because since there's proteins attached to it, we know that proteins are made by ribosomes. So if the rough endoplasmic reticulum has ribosomes attached to it, then we know that it's a protein factory, right? Now, the smooth endoplasmic reticulum does not have ribosomes. So guess what it does not make? Right, it doesn't make proteins. So what does it make? It's going to make lipids. It's going to make steroids. It also helps to detoxify the cell. So it does a few different things. And what we see here are these little cisternae. So what in the world's a cisternae? Have you ever heard of a cistern? Okay, you might not have heard of one, unless you grew up on a farm, maybe. But basically, on a farm, or I used to work at a place called the Morton Arboretum, and underneath one of the greenhouses, where we were doing some plant research, there were these huge cement tanks. And when it rains, these tanks fill up with water, and then you can use that water for watering the plants. On farms, sometimes they would do that. Use those cisterns, those tanks, to water crops, or sometimes they might even use it for drinking water. Okay, well, these are tiny cisterns, so that's why they're called cisternae. And again, they're going to be fluid filled. Okay, but it's the interior spaces that are isolated from the rest of the cytoplasm. Okay, the Golgi apparatus, Golgi body, Golgi complex, same thing, different names. It's for modification, packaging, distribution of proteins and lipids for secretion or internal use. So it's kind of like the UPS store of the cell. What can Golgi do for you? Right? So what it'll do is it'll take the raw product from the rough endoplasmic reticulum, and it'll take that and it'll modify it and turn it into a protein that can be utilized. And how we recognize this, to me it looks like a flat stack of pancakes. So it's flattened membrane stacks stacked on each other. So let's look at that. So here's our rough endoplasmic reticulum. It's making a certain protein, packages it up into a vesicle, ships it off. So it almost looks like a little UPS truck here. Okay, so then it combines with the Golgi apparatus, because remember, these are phospholipid bilayers. They're like soap bubbles. They can combine. So it pinches off, you get a bubble. This bubble combines with the Golgi apparatus, dumps that protein inside the Golgi, and now we can take that protein and modify it and package it up and ship it out to where it needs to go. Okay, so maybe it makes a vesicle inside the cell, and maybe the protein that's put inside that vesicle is an enzyme, like lysozyme. Well, if it's lysozyme, what did we just make? We just made a lysosome. Okay, we'll be talking about what lysosomes are in just a little bit. I've mentioned them earlier. Or maybe it just made a digestive enzyme, in your pancreas. And so it pinches off that little vesicle that has that pancreatic enzyme in it, attaches to the cell membrane, and through exocytosis, it kind of spits that out into a duct. Or maybe we can have a protein that's incorporated into the membrane here. Remember some of those cell markers? Well, it can make a vesicle to actually incorporate into the membrane to either repair the membrane or add a, one of those cell markers. All right, so that's Golgi. Okay, here's our lysosome. Remember, that's the digestive organelle within the cell. Don't confuse it with our digestion. But this is cellular digestion. And again, if a cell brings something in this case, it looks like pinocytosis. Remember that? It could be phagocytosis. Not this particular picture, because remember, phagocytosis has those pseudopods, those false feet that kind of grab, the microbe or whatever, and then pull it in. This just looks like we get what's called invagination of the cell membrane, and it pulls in this liquid that has some things in it. These could be microbes, they could be nutrients, what have you. Pinches it off into a vesicle. The lysosome attaches to that vesicle, dumps its enzyme, the lysozyme, into that vesicle, breaks down and destroys whatever is in there, and then we have this residual body here that can then attach to the cell membrane and expel that waste product through exocytosis. Another thing that lysosomes do, lysozyme is the enzyme inside, but another thing that lysosomes can do, if a cell is weak or damaged or dying, it can go into self-destruct mode, and this lysosome can break down and dump its contents into the cell to purposely kill that cell. Okay, so that's one way of taking out weak or damaged cells. Now, after an organism dies, this is part of what causes the breakdown of cells within the body. Besides bacteria, this is one of the first things that start breaking cells down, is that lysozyme action of these lysosomes breaking down and releasing that enzyme. So again, it's kind of a self-destructive type of mechanism to start decomposition. Okay, peroxisomes and proteasomes. Peroxisomes are smaller than lysosomes. They contain enzymes that break down fatty acids and amino acids. And hydrogen peroxide is a byproduct of that breakdown. Now, hydrogen peroxide is damaging to cells. I know you're probably saying, but I've used hydrogen peroxide on cuts and such. If you pour hydrogen peroxide onto a cut, what happens? It foams up, right? That's because we also make peroxidases, enzymes that break this hydrogen peroxide down, because it is kind of a toxic substance. Okay. And studies show, and this is the one thing I remember from my wounds class in grad school, was where they looked at cell healing, cellular healing after using hydrogen peroxide, and it actually slows healing down. Like if you keep pouring hydrogen peroxide onto a cut, it's going to take longer to heal because you're damaging these new cells that are trying to divide and fill up that cut. So it's not bad to use it initially because you're trying to kill bacteria. That foaming action also helps to disrupt any dead cells and dirt and what have you that's might be in a cut. But after that, you want to switch over to like a triple antibiotic cream, which actually speeds up the healing process. So, but again, hydrogen peroxide, not good to use all the time, creates oxygen-free radicals, which are damaging to cells. Okay, and then we have proteasomes, and they consist of large protein complexes, and they include several enzymes that break down and recycle proteins in the cells. Okay, the mitochondria, if you remember I said in that one picture, they reminded me of those marshmallow circus peanuts that are all sugary and everything. And what does sugar give you? Energy. Well, the mitochondria is the energy factory of the cell, provides energy for the cell. And that energy that it provides is ATP, adenosine triphosphate. So this is where it's made. And it has a couple of membranes. It has the crista, which are these folds of the inner membrane. And why do you think we have these folds? Any idea? Why isn't it just one smooth membrane? Well, it's to increase surface area. I kind of mentioned that. As a matter of fact, if I ask a question and you're not sure the answer, just say increases surface area. At least for the first semester here, the first few chapters, you'll probably be right. Okay, so basically it increases surface area because what we find on these cristae are enzymes. Enzymes like ATP synthase, which helps to make the ATP. And then we have the matrix, which is the substance located in the inner space formed by the inner membrane. Okay, so here's the matrix. And then the nucleus. And as you can see, we have the nucleus. We have the nuclear envelope. Okay, and envelope's a good name for that because we don't have just one phospholipid bilayer. We have two phospholipid bilayers. And I wish this were a little more magnified. But we have one phospholipid bilayer here and another phospholipid bilayer here. So a double phospholipid bilayer. And then we have these little pores here that are called nuclear pores. Okay. And this is where RNA, which we'll discuss later, but that can enter and leave the cell, or the nucleus, I should say. And so again, the nuclear envelope, it separates the nucleus from the cytoplasm and regulates movement of materials in and out of the nucleus. And then we have these little squiggles here. This is chromatin. It condenses to form chromosomes during cell division. So this is actually DNA. If we take DNA and we wrap it around some proteins called histones, and I'll show you what that looks like in a little bit, but wrap it around proteins called histones, and now it's a little more condensed. We call it chromatin. Now chromatin we still can't see under a light microscope. You need an electron microscope to see that. Same thing with DNA, way too small. You need a really strong electron microscope to see that. But when it condenses down more, because it's like, think about DNA like a thread. If we take that thread and we wrap it up and make yarn, yarn's a lot thicker than thread, right? And then if we take that yarn and we knit it into a sweater, well, that's even thicker, right? So that would be like a chromosome. And chromosomes we can see under our light microscopes. Okay. And so what else do we have here? Then we have the nucleolus in the center. And that's the assembly site for large and small ribosomal subunits. So in other words, ribosomes are made in the nucleolus. Okay? So ribosomes are made in the nucleolus. And what do ribosomes make? They make proteins. Okay, here's what I was talking about before, where you have a single thread, so that's your DNA. Take that DNA, wrap it up around these proteins called histones. Again, that's going to make it thicker, and so we call that chromatin. So it condenses it. And then, so that's like taking a thread, turning it into yarn. Now it's thicker. Now we take the yarn and we knit it into a sweater. Here's a fuzzy little X sweater. Okay, now it's a chromosome. That is so much larger, we can see that under the microscope. Okay? And then what this is showing is DNA is a double helix molecule. And what that means is it's shaped like a ladder, but you've taken that ladder and you've twisted it. And the rungs of the ladder are going to be made up of complementary nucleotides. And so like this red here is thiamine, and the what color is that? Blue, I guess. The blue is adenine, and then here the yellow is cytosine, and the green is guanine. So the best way to remember is A and T always combine. Okay, they are paired up. A pairs up with T. Adenine always pairs up with thiamine. If you can remember that, then if I say what does thiamine pair up with? Oh, well, it pairs up with adenine because A and T go together. Well, what's left over? The G and the C, the guanine and the cytosine. So they must pair up. Okay? So just remember A and T pair up, and you can extrapolate the rest.